So, guys, we talked last time in the first class about the cell adaptation, and we talk about what does it mean cell adaptation and how the cell can adapt with different stimulus and what are these stimulus. And we say that we have reversible cell adaptation and irreversible cell adaptation, and we talk in details about the difference between reversible and irreversible cell adaptation. And last class we talk about the cell injury. And last time we talked about the definition of cell injury, the types of cell injury, and we said that we have two types of cell injury: reversible cell injury and irreversible cell injury. And we talked about the mechanism of cell injury, how the cell can get injured, and why when we can say that this is reversible cell injury, and when we can say that this is a reversible cell injury, and what's the difference when the reversible cell injury can convert into a reversible cell injury. And of course, reversible cell injury means that the cell cannot the cell in reversible, the cell can come back to the normal structure if we delete the stimulus, so the cell will come back to the normal structure. But in irreversible cell injury, unfortunately, the cell will not be able to come back to the normal structure. So the cell will start to die off. The cell will die. Okay, when we when we can see that this is irreversible cell injury and when we can see that is irreversible cell injury. Let me repeat the question in a different form. What are the features of irreversible cell injury? Let me repeat the question in different form. What are the points of no return? No return. So the cell will not be able to return to the normal structure. What are the points of no return? What are the feature of irreversible cell injury when we can say that this injury is unfortunately irreversible cell injury? Anyone knows? So anyone knows the points of no return? The points of no return. When we can see that this cell cannot come back to the normal structure. This cell is gonna die. So anyone can write in the chat or let me open the mic. So anyone know the point of no return? Guys, the mic is open. Try with me. Just think and try with me. I don't want to choose any student, so Unfortunately, I have to ask you one by one. So first one, Hamza bin Abid. Hamza, open the mic, please. Ah, he left. So he's gonna be removed from the group. Adnan also, he's gonna be removed from the group. Okay. Adnan and Hamza, they will not be removed from the WhatsApp group. They are not, they will not be available anymore in our class. So the points of no return, when we can say that the cell will not be able to come back to the normal structure. If one of three structures are damaged. One, of course, cell membrane. Cell membrane. Two, mitochondria. Three, nucleus. The cell cannot live without any one of these organelles. Of course, if the cell membrane is damaged, so we don't have cell anymore. We don't have any cell because the outline of the cell is damaged. And of course, the cell cannot live without mitochondria because the mitochondria is the factory of energy inside the cell. And also, when we said last time, when if the mitochondria is damaged, a lot of free radicals and a lot of enzymes will be released and start to damage all organs and all cells. And of course, the nucleus, because without nucleus, without DNA, so the cell cannot live. So the points of no return, if we lost one of the most important structure in the cell: cell membrane, mitochondria, and nucleus. Cell membrane, mitochondria, and nucleus. So if these three organs are intact, if they are intact, and if they are not damaged, if they are okay, so the cell can come back to normal structure. But if one of these organelles is damaged, so unfortunately, this injury is irreversible cell injury. Okay, we have two types of reversible cell injury. Two types of reversible cell injury. First one is hydropic degeneration, and second one is called hyalinosis. So reversible cell injury we have two types of reversible cell injury. First one is hydropic swelling. Hydropic swelling. So second one is hyalinosis. Both of them, they have the same concept. I mean that hydropic swelling, hydro means water. Hyalo means hyaline protein, some protein. So hydropic swelling, when the cell, the cell and the nucleus, when the cell start to be accumulated with water, and the cell, when the cell lost the ability to release the water. And here, when the cell, sorry, when the cell is accumulated with proteins, which senses, and the cell lost the ability to release the protein. So hydropic swelling, when the cell start to accumulate a lot of water and lost the ability to release this water. And we will talk now about the causes. And here when the cell start to accumulate proteins inside the cell and lost the ability to release these proteins. And of course, proteins or water will start to compress on the organelle and compress the organelle and start to injure the organelles. And this water or protein start to injure the organelles. But is it reversible? Of course. If we delete the cause, so this water will be released outside and the cell will come back to the normal structure. And of course, here, if we delete the cause which lead the cell to accumulate the protein, so the cell will release the protein outside and the cell will come back to the normal structure. So let's start with hydropic swelling. Hydropic swelling. What is the cause of hydropic swelling? We mentioned last time the causes. Do you remember the cause of any cell injury? The main factor is hypoxia. And we said if the cell get hypoxia, so we don't have enough oxygen, so the cell cannot produce enough ATP. And what the problem? We said last time that we have here some pump, it's called sodium, potassium, ATPase pump. This pump use ATP, 1 ATP to release sodium outside the cell and get potassium inside the cell. So now we don't have ATP, so this pump will not work. So the potassium will stay outside and sodium will be accumulated inside the cell. And we said that sodium always bring water with it. So a lot of water will start to accumulate inside the cell. But if we could delete the cause, the cause which lead to hypoxia, and we said the main causes of hypoxia is ischemia or anemia. So ischemia means that the blood cannot go to the cells. So of course, there's no oxygen going to the cell. So if we treat ischemia, so we don't have hypoxia anymore. So we have a lot of ATP, then the pump will start to work and release sodium outside, and water will follow the sodium outside, and the cell will come back to the normal structure. So here, the hydropic degeneration, we say that it's reversible cell injury with intracellular water vacuoles, water accumulation inside the cell. But it's reversible if we delete the cause, so we can solve the problem and the cell will come back to the normal structure. And Of course, the main cause is hypoxia. The pathogenesis we talked about it last time. We said hypoxia leads to loss of ATP. ATP, there's no ATP, so the sodium pump will not work. The sodium will start to accumulate inside and will draw the water inside the cell, and the cell will start to get swelling and accumulate a lot of water. But if we can delete the main cause, hypoxia, so we will get rid of cell swelling. We will delete this pathogenesis. So, but it's reversible until the water can damage any one of these compositions, because in hydropic swelling, the water will accumulate in any organelle, but not in mitochondria, not in nucleus, and the water will not damage the cell membrane. Because if the water damage one of these organelles, so we cannot say that it's reversible. So it converted into irreversible. So hydropic swelling, it's reversible until the water can damage any one of the three compositions: nucleus, mitochondria, or cell membrane. So the the site of hydropic swelling always occur in specialized organs, organs which are used in metabolism, like liver or kidney. So it occurs more in liver and kidney, because we know in liver we have a lot of metabolic process. We use a lot of ATP. And in kidney also, we use a lot of ATP to filter the urine. So hydropic swelling or hydropic degeneration occurs in highly specialized the organ which use a lot of ATP to make a lot of metabolic process. The gross, the gross biopsy, if we look at the if we look at the organ, so we see that the organ is big in size or small. Of course, it's big because it's swelling, contain a lot of water, so it's swelling and big in size. The color, and it's important. The color of the organ is pale. It's not it's not good color, or it's not heavy color. It's pale color. Why it's pale? Because here the capillaries, which is going inside the cell and supply the cells with blood. Here we say that we have a lot of water accumulated. A lot of water are accumulated. So this water will start to compress these capillaries, blood capillaries, so the blood cannot go probably to the cell. So the cell will start to be deprived from blood. So the cells will start to get pale color, because the water which is accumulated inside the cell start to compress the blood capillary. So the organ, we have to know three things: that the organ is larger in size, swelling, mean contains water, pale, and soft. Of course, soft because it contains water, so it's not hard. So it's soft because it contains a lot of water. Larger in size, swelling, pale because of compressed capillaries, and soft. If we look at the cells in by microscope. So microscopic examination, we will see the cell is swelling, but the membrane is intact. It's not damaged, because if it's damaged, so we are not talking about reversibles. So now it will be converted into irreversible. But here we are talking about reversible stage. So intact membrane, it's intact. Swelling organelles, especially endoplasmic reticulum. But the mitochondria is not swelling, because if mitochondria is damaged, so now we are not talking about reversible. So it will be converted into irreversible. So the cell, of course, contains a lot of water, so it's swelling, large in size, but membrane and mitochondria are intact. The nucleus is intact. They are not damaged. And swelling organelles, especially endoplasmic reticulum. And stroma means the fiber around the cell, around the organ, the fiber. This stroma outside contains blood capillary. So we say, of course, this compress on blood capillary. So this trauma will be pale in color, pale in color. So stroma compressed contains compressed capillary, which lead to get pale color of the organ. So if we use more stronger microscopy, it's called electro electron microscopy. So of course it's more stronger than normal microscopy. So we'll find the cell like this shape. So let's describe each thing of the cell. Of course, the cell is swelling, a lot of water. So the cell is swelling. Water vacuoles, a lot of water vacuoles. What is this? What is this? The normal cell, this is the normal cell. And when the water starts to accumulate here, so of course the water will compress the cell membrane, and the water will accumulate here. But the cell membrane is intact. It's not damaged. It's not damaged. If it's damaged, so it's not reversible. But now the cell membrane is intact. It's not damaged, but it's compressed, because the water will move some part of the cell membrane, and it's called blips. Some vacuoles on the cell membrane, which which are accumulated with water. The normal epithelial cells, we know that the normal structure of the normal epithelial cells like this. The cell membrane contains this thing which called microvilli. When the water accumulates here, so of course the water will delete these microvilli, because the water will move the cell membrane. So there is loss of microvilli, loss of microvilli. So first, of course, the water is accumulated, so the cell is swelling. The water will move some parts of the cell membrane and form something called blips. And the water will move the microvilli and cause loss of microvilli in some epithelial cells. And in normal structure of the cell, we have something called endoplasmic reticulum. Endoplasmic reticulum. So this is the cell, and this is the nucleus, and here we have something called endoplasmic reticulum. And we have two types of endoplasmic reticulum: smooth and rough. Rough, why we call it rough? Because it contains some granules on the top of the endoplasmic reticulum. It's called ribosomes, which help in synthesis of protein. So this is rough endoplasmic reticulum. Rough because it contains a lot of ribosomes are attached to the surface of rough endoplasmic reticulum. But when the water accumulated here, when the water accumulated, so the water will start to move these ribosomes from the rough endoplasmic reticulum. So there's so the ribosomes will not attach to rough endoplasmic reticulum anymore. So there is detachment of ribosomes. So first of all, the water will accumulate in the cell and cause cell swelling. So let's repeat again. So the water will start to accumulate in the cell and cause cell swelling. Then the water will move some parts of the cell membrane and form something called blebs. Then the water will start to move the microvilli in some epithelial cells and cause loss of microvilli. Then the water will start to move the ribosomes from the rough endoplasmic reticulum and cause detachment of ribosomes. So cell swelling, blebs, and loss of microvilli, and ribosomes are detached from the rough endoplasmic reticulum. And of course, the endoplasmic is swelling, and also here mitochondria is swelling. But you told me that in reversible the mitochondria cannot get water swelling. No, mitochondria is very slightly swelling because electron microscope is very strong, so it will show very slight water inside the mitochondria. But most of the water are getting inside other organelles like endoplasmic reticulum. And inside the nucleus also very slight amount of water will get inside the nucleus and compress the chromatin and the chromosome together and cause something called chromatin clumping or condensation. But they will not they will not damage the nucleus or mitochondria because they are very slight amount of water inside the mitochondria and the nucleus. But if the water amount is increased inside mitochondria or nucleus and causes damage of these organelles, so of course the cell will die because the cell cannot live without mitochondria, nucleus, or cell membrane. So the water will affect cell membrane, mitochondria, and nucleus, but very slight amount. Here, some blips, but the cell membrane is intact. It's not damaged. Loss of microvilli, but it's still intact. It's not damaged. The mitochondria, very slight water inside the mitochondria, but still intact. It's not damaged. The nucleus, very slight water amount inside the nucleus, but it's intact. It's not damaged. So the water will just cause some chromatin clumping. Clumping means the chromatin will be condensed. They will attach together, but they will not be destroyed. So it's still reversible. Just remove the cause of water swelling and everything will come back to the normal structure because it's reversible. But if mitochondria is damaged, nucleus is damaged, or cell membrane is damaged, so of course, and unfortunately, it's gonna be irreversible. But till now, it's reversible. Just if the cause is removed, cell return to the normal structure. But if not, so the cell will die. Necrosis of the cell, death of the cell. Some clinical pathology on hydropic swelling. You study the normal structure of the small intestine. The small intestine epithelium is like the microvilli. This microvilli, this surface is called the brush surface of the small intestine. And this brush surface, this microvilli contains a lot of enzymes and secrete a lot of enzymes which help to digest and absorb nutrients and also help to absorb the water inside the bloodstream. So if these microvilli are lost, if they lost it, so of course the water will not be absorbed inside the bloodstream, and the water will start to accumulate in the lumen of the intestine, and this water will be released from the colon, then from the anus, and cause watery diarrhea. Diarrhea means a lot of water with feces. So the feces will be very swelling and watery because the water are not absorbed from the microvilli because microvilli are lost. We can also get maldigestion and malabsorption, deficiency in absorption and digestion, because these microvilli were containing a lot of digestive enzymes and a lot of substances which help us to absorb the nutrients. So if these microvilli are lost, so we cannot absorb or digest nutrients and we cannot absorb the water. So we will start to get maldigestion, malabsorption, and water diarrhea. Also in liver, if the water is accumulated inside the liver, so the liver will start to be big in size and get larger and larger, and it's called hepatomegaly. Megaly means enlargement of the liver side, and of course this can cause something called jaundice. Jaundice means accumulation of yellow pigment. It's called bilirubin. Bilirubin is supposed to be secreted out of the body. It's a yellow substance. So the liver helps us to remove this substance. If we have hepatomegaly, hydropic swelling of the liver, so we cannot get rid of this substance. So this substance will accumulate in your body and start to cause a dangerous disease called jaundice. And of course, we will study jaundice in details. So again, so hydropic swelling, it's reversible cell injury caused by accumulation of water. The cell will lose the ability to remove the water out of the cell. Why? The causes we say that the main cause is hypoxia. Hypoxia can lead to loss of ATP. Loss of ATP leads to failure of sodium potassium pump. The sodium will be accumulated inside the cell. Then the sodium will draw the water inside the cell. The cell start to get a lot of water and filled with water and cell swelling. Cell will start to compress the organelles and get inside the organelles and cause organelle swelling, except mitochondria, cell membrane, nucleus. Because of the water damaged one of these three things, so now it's not reversible, it's irreversible. So we talk about the gross picture. Gross means with your eye, you can see that the organ is big in size, of course, swelling, contain a lot of water. It's pale. Why? Because the water will compress the blood capillary and the blood will not draw to the cells and the cell will not be supplied with water. So the organ as a whole will be pale in color. And of course, it's soft because it's water. Microscopic examination, if we describe the cell under the microscope, the cell will be swelling, but with intact membrane, intact mitochondria, and intact nucleus. And of course, there are some water vacuole. There are some water accumulated inside the cell. And stroma. Stroma means the outlines of the organ which contain the blood capillary. The blood capillary in the stroma will be compressed, which lead to pale color of the organ. And under the electron microscope, which is stronger than normal microscopic, the cell will be swelling. The water will push the cell membrane and cause two things: blebs and loss of microvilli. And the water will push the ribosomes from the rough endoplasmic reticulum and causes ribosome detachment. And the organ will get swelling like endoplasmic reticulum, and the nucleus and mitochondria will be very slightly accumulated with water. Very slight amount of water inside the mitochondria and inside the nucleus, and the water inside the nucleus will push the chromatin and causes chromatin clumping. And the fate, you have two options. First option, eliminate the cause, and then the water will be released. If we relieve the hypoxia, then you will get good amount of ATP, so the pump will work and will take outside the sodium, and the sodium will take with it the water outside, and the cell will get rid of and release the water and will come back to the normal structure, because now it's reversible, because now it contains mitochondria which can produce ATP and solve the problem. But if you don't treat this pathology, so the water will start to accumulate and accumulate, then get inside the nucleus and get inside the mitochondria and start, these blebs start to be ruptured and the cell membrane will be damaged, and now unfortunately it's not reversible, it converted into irreversible cell injury. And clinically we said if it happens in small intestine, will lead to loss of microvilli. These microvilli were very important in absorption and digestion. So there's no good absorption So we have malabsorption syndrome, and there's no good digestion. So we have maldigestion syndrome, and the water will not be absorbed. So the water will be in very, very big amount in colon, which can cause to watery feces and watery diarrhea. And in liver can lead to hepatomegaly, and this hepatomegaly can cause very dangerous disease called jaundice. And of course, we will study jaundice in details later. And the second type of reversible cell injury is hyalinosis. Page number 17. Yeah, here. It's page number, no, sorry, 16. Page number 16. Hyalinosis. Hyalinosis, it's also accumulation, but not the water. Accumulation of proteins. Some cells, they are normally produce protein, but now they cannot release this protein, which they synthesize, and the protein will start to accumulate inside the cell and compress the organelles. So here it's microscopic change in tissue proteins. The protein will be accumulated and became homogeneous. What does it mean homogeneous? And what does it mean eosinophilic? Eosinophilic means red or pink color under microscope. It's easy because we know in microbiology that the normal color of protein under microscope is red. So the cell will be in red in color or pink in color. Why? Because accumulation of the protein. But what does it mean homogeneous? Homogeneous means structure less, loss of the normal structure. Why? There's the cell, and here the nucleus, and here proteins. This is the normal structure of proteins. But when these proteins start to be accumulated, so when the cells start to accumulate, proteins start to be accumulated. So this is the normal structure of protein? Of course not. Of course it's not. So the protein start to lose the normal structure. So it's called homogeneous or structureless. So the hyalinosis, accumulation of proteins or microscopic change in tissue protein, the proteins are not released anymore, then they accumulate together and start to lose the structure and start to be homogeneous. And of course, eosinophilic means under the microscope, it's red or pink or rose in color. The causes of hyalinosis, we have extracellular hyalinosis and intracellular hyalinosis. Of course, extracellular means outside the cell, accumulation of proteins, but outside the cell. And intracellular hyalinosis, it's accumulation of protein inside the cells. Let's start with intracellular hyalinosis. 4P. First P, some blood cells, which is P, plasma cells. And we know that plasma cells produce antibodies. Magid. Do you know Magid? IgM, IgA, IgG, IgE, IgD. They are proteins in structure. They are proteins. The plasma cells, for any cause, the plasma cells will start to lose the ability to release these antibodies. We know, of course, the main cause is hypoxia. So the cells now lost a lot of ATP, and then the cell doesn't have a lot of energy, so the cell cannot produce the proteins which synthesize normally outside the cells. Then these cells will start to accumulate together inside the plasma cells and compress the organelles and cause and some red bodies, because we know the protein are red or pink in color, accumulate inside the cells, and we call it Russell body. The Russell bodies, it's the antibodies which are accumulated inside the plasma cells. Second example of intercellular helinosis, parenchyma of liver. The liver is the most affected organ due to alcoholism. When you drink a lot of alcohol, the first organ which will be affected is the liver. And we know, of course, that the liver synthesizes a lot of proteins, because we know 99% of metabolic process occur in liver. So the liver synthesizes a lot of protein. So when you start to drink a lot of alcohol, so a lot of alteration in metabolic process will happen inside the liver, and the liver will start to lose the ability to release the proteins outside the hepatic cells, the liver cell. And these proteins will accumulate inside the hepatic cells, and these deposits are red in color, and it's called Mallory body. So Mallory body, it's protein accumulation inside the hepatic cells or the liver cell. Hepatic means liver. The third example is P, proximal convoluted tubules. In kidney, we have nephron, and the nephron this part is called glomerulus, and this is proximal convoluted tubule. Proximal means near, and this is the distal. Distal means far from the glomerulus, and between of them lobe of Henle, and this is called collecting duct. And you know that the urine pass through these tubules, and the urine, before get inside the nephron, it's called primary urine. And the primary urine, it's not the urine which you secrete out of the body. Why? Because this urine contains a lot of important things: proteins, water, glucose, sodium. So we need to convert this primary urine into secondary urine. This urine which is released outside the body. So to convert the primary urine to secondary urine, you have to go through three main processes: filtration, reabsorption, and secretion. Filtration means there are two things will not pass and will not will get inside the tubules: red blood cells and the proteins, because they are big inside, so they cannot go from the the holes of the glomerulus. So they will come back to the body because we need them. Of course, normally you don't have blood inside your body, inside your urine. You don't have protein in your urine. Then in plasma tubules and in all these tubules, you have two main process. Reabsorption means some substance from the water will get inside the bloodstream. We will get back to the blood. And secretion means something from the blood we will put it inside the urine. We will say, sorry urine, you forget to take these things. Please take it. So in some diseases in kidney, the protein, some disease can affect the glomerulus, and the protein will pass through the tubules. Then the proximal tubule will see the protein. Wow, why the protein get inside the tubule? It's supposed to get filtrated and doesn't pass through the tubules. So proximal convoluted tubules will start to reabsorb the protein, which is not normal because the proximal convoluted tubule cells cannot deal with the protein. It's not normal to reabsorb protein. So the cells of proximal convoluted tubule will reabsorb the protein, and of course the protein will accumulate inside the proximal convoluted tubule cells, and the cells cannot treat or deal with these proteins, so the cell will not be able to release these proteins and start to form something called some red deposits inside the cells. It's called hyaline bodies. Hyaline. So in plasma cells, Russell bodies. In parenchyma of the liver, Mallory bodies. In proximal convoluted tubules, it's hyaline bodies. The last P is pneumonia or diphtheria. Pneumonia happens due to some infections. Some bacteria can lead to infection and inflammation of the lung and cause pneumonia. And diphtheria also, it's bacterial infection. These bacteria release some toxins inside the body, which lead to toxemia. Emia means blood, so toxicity in the blood. These toxins can travel to a lot of tissues, and we will talk in separate chapter about toxemia and septicemia. One of the most important organs which can get a lot of toxin is skeletal muscles, because you know skeletal muscle needs a lot of blood supply, and the blood now contains a lot of toxins. So a lot of toxins will get inside the skeletal muscle. So the skeletal muscle will not be able to work normally, and the skeletal muscle will not be able to use the oxygen because this toxin will start to destruct or damage the channels inside the skeletal muscle cells. So some dangerous substance will start to accumulate inside the skeletal muscle. It's called lactic acid. Lactic acid. And of course, lactic, it's acid. So it leads to acidosis inside the skeletal muscle cells. And acidosis can lead to something we will study in biochemistry. It's called denaturation of proteins, breakdown of normal proteins. So this acidosis will lead to breaking down of important proteins inside the skeletal muscle cell, and these proteins will, when be damaged, they will start to accumulate together and form pink or red striation, some line, red line. It's called Zenker's degeneration. Zenker's degeneration. Zenker's degeneration. So in plasma, Russell body, in parenchyma of the liver, Mallory body, in proximal convoluted tubules, hyaline body, pneumonia or diphtheria lead to toxemia and accumulation of Zenker's body inside the skeletal muscle cells. So we're gonna stop to this part. So next time we'll start to discuss extracellular cyanosis and mycosis and mycoid changes. Then we will go to irreversible cell injury. I hope you understood the lecture. So anyone has any question? So guys, to be serious,